Hello, welcome back. This is topic 2.3, and this topic is all about carbohydrates and lipids, which are basically energy storage. So here are the aims, understanding, applications, guidance, everything IB expects you to know about this unit. If you want to take a moment and look at that. So first things first, monomers to polymers. This is not just a topic that kind of is not limited to this subtopic alone. It actually branches over every single molecule that we're looking at, but basically we have to look at the process at which we go from a monomer to a polymer. So first things first, we have to figure out what are the monomers of carbohydrates. So basically, for carbohydrates, we're going to be talking about monosaccharides. So for instance, just this one here, or this one here. If you'll notice, these are both alpha glucose. Just kind of reminding you should be able to differentiate between alpha and beta glucose and what they're used for. Alpha glucose is more starch, which makes it because it's a little weaker, looser structure, while beta is more rigid, and this is for cellulose. So aside from that, we should know the monomers. These monomers are going to go through reactions, as we mentioned in the first video, topic 2, and it's going to create polymers, which are known as disaccharides. And if you put many disaccharides together, what you're going to end up with is polysaccharides. So now, just a little quick review. When talking about reactions, there are two types of reactions. There are catabolic reactions and anabolic reactions. For the main chunk of this, we'll be talking about anabolic reactions, which is condensation synthesis reactions or dehydration reactions, because we'll be talking about how there basically comes out a water. It's basically we're dehydrating the molecule, we're condensating a water molecule in order to create a bond between these two molecules. So if we just look right off the bat, since we talked about, if you remember the structure of a carbohydrate or a sugar molecule is going to be kind of double oxygen, or oxygen and carbon at a similar ratio in a ring or linear structure. So looking at the first one, this kind of makes sense. This kind of looks like a familiar a monosaccharide that we're familiar with. Um, so this is glucose. So maybe this is probably the answer. If we go here, we're going to look at this three-ringed carbon, and you notice that there are absolutely no, I said three ring, I mean it's just a ringed carbon structure, and you'll notice that there are absolutely no oxygen. Since we have none of those oxygen, it's not going to be correct. And then we'll go off onto this C. If you look at this one, this is going to, it has the oxygen, and it has that, they're kind of similar ratios, but we're going to look at basically this R group. When you see an R group, immediately think amino acid. And now this, you may be asking, well, this also has a string of carbons with hydrogens attached. But if you'll notice, these oxygens are nowhere near similar to the ratio of carbons. So basically, it's nowhere near 1-to-1 ratio, and we're left with A. So branching off from that monomer, we're going to have three main monomers that IB wants you to know. This is going to be glucose, fructose, and galactose. These are basically, you're not going to need to know the structure of each one individually, but you'll need to know these are the monomers that basically make up most of the sugars that we'll be talking about throughout the course. So now, when we go to from monosaccharides to disaccharides, through that condensation, dehydration, whatever you want to, anabolic, all are correct terms, reaction, basically we're going to get three different types of disaccharides. So when we combine two glucose together, we're going to get maltose. When we combine glucose and fructose, we're going to get sucrose, which is also table sugar. And if we combine glucose and galactose, we're going to get lactose, which is a common sugar found in milk. So, after looking at all of these, basically now what we can see is that, or basically, you're going to want to have to memorize all these and basically just kind of get these under your belt. So here's an example of how you might be tested on this. Which sugars are examples of monosaccharides and which sugars are examples of disaccharides. So give me a second. Okay, so if you can quickly list off our monosaccharides, we have fructose, galactose, and glucose. So lactose is not a monosaccharide, it's going to be a disaccharide, and so is sucrose. So both B and C are wrong. And if we look at the second column, on our answers left, we have galactose and lactose. Well, galactose is a monosaccharide, so it's going to be D. So now, once we have we go from monosaccharides to disaccharides, and now we're at polysaccharides. There are three different categories IB expects you to know with this. We have cellulose, starch, and glycogen. Cellulose and starch are made by plants, as you can see here, while glycogen is made by animals. As I mentioned before, this beta glucose is important because of its structure. We get cellulose more rigid, while for alpha, all these alpha glucoses, we get starch and glycogen, which are both energy storage. And since this alpha structure gives them a not so compact structure, as you see, this is very extremely compact. This is not so much compact as you see over here. So we basically can understand, or this is basically the reasoning behind why this is used more for energy storage, because it's easily broken down. So this is kind of a chart because it's kind of hard to explain all these things individually, and I think this chart really puts it all good together. So if you want to copy the chart, take a picture or something, this is really useful, and you'll kind of want to not per se memorize all these things, but you kind of want to know the properties of each thing. You want to know that cellulose does not branch, and you kind of, when you think about cellulose, it's a very compact, organized structure, so you can kind of get that there's no branching here. Now, when you go to starch, you're going to need to know the difference between amylose and amylopectin. You're not going to really need to know the functions, the differencing in functions so much, but you'll really need to know kind of like the difference, as in amylose does not have a branching structure while amylopectin does. And because of this, we have 1 to 6 bonds. And now if I draw a simple glucose molecule real quick, how we label carbons is basically, this is going to be your generic. You'll start on the first carbon in the ring. This will be like an oxygen. So you have 1, 2, 3, 4, or 5, 1, 2, 3, 4, 5, 6. So basically what you'll notice here is that, so you'll have the 1 of one molecule is bonding to the 4 of another. And that's kind of what it's talking about. And if you have 1 to 6, it'll be a 1 to a 6 over here. So going kind of away from that, we kind of see that the similar between glycogen and amylopectin is that we have branching structures. And what you'll need to know between these two is that glycogen just has a very more sporadic and much branchier structure, I guess you could say. And another thing you might want to know is that amylose is used more for plant storage, and it's harder to digest, but it takes up way less space. So here's an example of a question you might be asked. What carbohydrates are used to provide energy storage in animals and plants? Well, going back, we know that starch is going to be plants and glycogen is going to be animals. So, if we look here, we have cellulose. We already know that this rigid structure makes it kind of really hard to store energy, so that's going to be a cell wall. In maltose, we didn't really talk about maltose in plants, so that's not going to be, it's going to be looking at starch. Starch is that key indicator of plants. And now, so we're left with A and C, so we're glucose and glycogen. Now, it is true that glucose is used in order for cellular respiration, which is topic 2.8, but this is also used for plants. It's more glycogen that they're trying to get you at because of starch versus glycogen, as we saw back here, the starch versus the glycogen. So we're left with C. Okay, so kind of moving away from carbohydrates, I know that was a lot, and this unit can definitely get a little confusing, so we're going to kind of just take a moment to pause and recap and refresh and get all carbohydrates, maybe go back and watch straightened out. But at this point, we're going to start talking about lipids. So lipids are, as we pointed out before, are similar in their carbon chain structure, but they all have hydrogens attached to them. So there's way less oxygens, maybe 1 or 2. The ratio is completely different. So if you see a little amount of oxygen compared to carbon, you're going to clear indicator it's a lipid. So now each of these blue bonds is a bond kind of representing a bond to a hydrogen. I just didn't want to draw them all out, and usually they won't depict the hydrogens, but for this purpose, I just kind of drew the bonds. So we have 3 different types of lipids of these chains. This is going to be a completely saturated chain. This is because every single carbon is surrounded by hydrogens. The clear indicator of this is single bonds. You will never find a double bond, a triple bond, in a saturated chain. These two are both unsaturated. Now, specifically when we have one double bond, or a double bond, real quick, is a clear indication that you're going to have an unsaturated, because that bond takes away a hydrogen from the carbons that it's bonding. As you'll notice here, they only have one on each side rather than two. So, when looking at saturated, if it only has one, it's going to be monosaturated. And if it has two double bonds, it's polysaturated. So, real quick, we have saturated, unsaturated, these two, monosaturated, mono-unsaturated, polyunsaturated. Now you'll see that there's two different kind of factors. So now that we have only two hydrogens surrounding these carbons, they can be put in different places. If they're opposite, like seen here, it's going to be straight. And when it's straight like this is going to be known as trans fats, which you kind of might hear a lot of these terms, might be familiar from basically like the media and social media and all these products saying trans fats, unsaturated fats, and we'll get kind of like into the health effects of each, but basically just know that trans scientifically means that the hydrogens are on opposite sides, so we're giving a linear molecule. However, when you have hydrogens on the same side, this is kind of affect the bonding, and you're going to get a bent molecule, and this is cis. This is known as cis fats. So now you kind of have all the terminology. You have saturated, monounsaturated, polyunsaturated, trans, and cis fats. So real quick, just kind of recap, the function of lipids is energy storage. It can be used for hormones, as seen in steroids. It can be used as insulation, which is fats. Protection, which is also kind of fats and waxes, and then structural, which is kind of the phospholipids, which we discussed in topic 1. So another thing that you're supposed to, IB expects you to know is triglycerides. So what is this? What this is going to be is one glycerol. This molecule here is a glycerol, and 3 hydrocarbon chains. As you'll notice that this is, if I asked you just this chain, this is going to be saturated, saturated, unsaturated, and this bent double bond is basically telling you cis. This is all made through the condensation synthesis reaction, anabolic reaction, as mentioned in the beginning. So here's a question that you might be asked. What characteristic shows that a steroid molecule is a lipid? So once you get this molecule, you'll be saying, okay, well, carbon, hydrogens might be it can be either a carbohydrate or a lipid, because I really don't see the R group or anything else indicating it's not a carbohydrate or a lipid. So made of carbon rings. Well, if you think back to glucose, well, that's a carb, and that's also made of carbon rings. No. As a very low proportion of oxygen to carbon. Now, this is a telltale sign. If you remember, carbohydrates are CnH2nOn. So basically, these oxygen and carbon are the same. So that's kind of maybe we'll come back to it. It contains a hydroxyl group, as do fatty acids. Now, this is true, but a hydroxyl group can also be found in carbohydrates, and it is made of nitrogen, oxygen, and hydrogen. Well, I don't see any nitrogen here, so that's just wrong. So we're going to be left with as a very low proportion of oxygen to carbon. So now we start talking about BMI, weight, health. Basically, this is going to be more of the health effects of lipids on, the human body. And when we start talking about this, I didn't really necessarily include a slide. I'm just going to go over quickly, because it's not so much testing you on these various things, and you can do more research on this on your own, but it's more trying to get you in the habit of starting to think of claims that are being made in the scientific process. And there'll be more questions. We'll start including more questions on how more focused on your ability to interpret data rather than strictly recite fact. So right off the bat, basically the claims that have been made is saturated fats and trans fats are bad, and they increase bad cholesterol, while cis and unsaturated fats are good. They increase good cholesterol. So the back in between this is there's something called a low density lipoprotein. And what this does is basically saturated fats and trans fats and all these so-called bad fats are increasing LDL. And what LDL does is it carries cholesterol from the liver to the body. So we're increasing that cholesterol. And basically what's happening here is we also have high density lipoproteins. So HDL and LDL. And high density lipoproteins, on the other hand, will harden arterial walls and form fat deposits, decreasing blood flow. So while so what happens is these cis fats will increase HDL. So we're basically increasing cholesterol. We're hardening arterial walls, and this is just going to cause a lot of bad problems on the body. So basically, the claims on health here is that saturated and trans fat increase the risk of coronary heart disease. Um, we'll talk about more coronary heart disease when we get to the heart, which is a later topic in topic 6. So basically, you just kind of want to know, again, this is more looking at the scientific process. So when you go further from this, you have to start looking at the claims, and basically what you'll have to do is you'll see kind of correlations between people having saturated fats and coronary heart disease, and you'll kind of go at claims against the saturated and trans fats decrease heart disease, just basically on the population you're looking at, and there's some conflicting evidence. And basically what this is asking you to do is when you're given a set of data, you're going to have to make claims and then look at not only the supporting evidence, but also the evidence against it. So I encourage you, if you're curious as to what exactly is going on with this whole trans fats, saturated fats, LDL, HDL, to kind of research that. And if you kind of want to understand more of the biology behind it, basically from this, you should be able to make an argument and support it, and also recognize arguments against it. So finally, we have BMI. Now this is a loose regulator of health by taking mass in kilograms over mass in height, or height in meters squared. And you're going to want to know how to calculate BMI if given any of these components, or how to calculate any of the components given BMI and maybe one other. And all at the same time, we should be able to look at a BMI chart and kind of see, okay, well, this is the normal weight here, green. So basically, this is trying to tell you, if you calculate BMI and it's within any of these ranges based on height and weight, then this is basically a good. This is a good place to be, and basically a low BMI means that you're underweight, a high BMI can lead to obesity, and basically that's all we really need to know. There's not too much in depth. So the final part of this topic is carbs versus lipids in energy storage. So basically, as I said in the beginning, all these carbs and lipids, their main purpose and their main function as we kind of start looking deeper into them is energy storage. And how we kind of get energy, how we cellular respiration, which is topic 2.8, and getting energy from them. So different factors that we look at is osmolarity. So this is, as we talked about, osmosis. If you don't remember, it's the movement of water molecules from a high concentration to a low concentration. If you have a lot of water molecules, lower water molecules over here. Basically, if you have carbohydrates dissolved, this is going to increase or change the osmolarity more. While if you have lipids dissolved, it's not going to really affect as much as carbohydrates this osmolarity, or this rate of osmosis. We have digestion, and when you have carbohydrates, think of food, bread. This is going to be more readily. It's going to be in a form that's really easy to use. While if you have lipids, this is going to think fats. Fats, not so much fats like you eat, but fats inside your body. You're going to have to go into reserves and break it down. ATP yield. You get half as much ATP per gram, and you get twice as much ATP per gram for lipids. So this is kind of showing that basically lipids are a much more concentrated form of energy storage, which kind of makes sense. The body kind of fine-tuned this process in order to make fat a much more efficient way of storing energy in case of times of low food. And then finally, we have solubility. Carbs are more soluble due to their slight polarity, while lipids are hydrophobic, as you can kind of see when you put oil in water. So here is a question that you might get on a test. Again, I wanted to point out, that this is not so much recalling facts and information, but taking the data and evidence and being able to draw conclusions. So I'll give you a minute. Okay, so the first question we get from this graph is describe using the triglyceride levels on the graph, the results of site 1 at site 2 for all the birds. So we're looking at triglyceride levels, so that kind of tells you we're looking at this graph. And then we're going to look at basically site 1, which is the darker bars, as we saw here, versus site 2. If you'll notice quickly, okay, well, all the site 1s are higher than site 2. And then you can also point out, well, this, I didn't include this here, but the hermit thrush, it'll give you the names of the birds. You won't, oh, here, sorry, they're right here. Kind of blind. So the hermit thrush has a higher triglyceride level than all the other birds. And we look at the lower triglyceride levels, you'll see, well, the American robin has the lowest for site 2, but the Mongolia warbler has the lowest for site 1. Explain the difference in triglyceride levels for the butyrate level and for the hermit thrush at site 1 and site 2. So for this one, you might be wondering, well, what's so much like, what's really that? What is butyrate? And basically, it'll give you some information down here where it's talking about how among birds, high triglyceride concentration in blood plasma indicates fat deposition, where this indicates fat utilization in fasting. So basically, you can look at here and you can say, well, at site 1 for the hermit thrush, no, here we're just looking at this one right here. At site 1, the triglyceride is high and the butyrate is low, while at site 2, the butyrate is much higher, and the triglycerides are low. So what this is telling you is that the hermit thrush at site 1 is most likely eating more. They're depositing fat, they're not so much utilizing fat as much, whereas at site 3 they have much higher butyrate levels, so they're more likely to utilizing that fat and not eating as much. Here's a kind of a summary of all the information. And since I don't want to go over time too much, here are a couple more questions that you can answer on your own, and I will give the answers in a minute. But basically outline the types of carbohydrates, so you're going to think the different types of saccharides, and giving those examples, the name examples that we pointed out, and describes the importance of hydrolysis and digestion. While you don't really you won't at this point, topic 2.3, you may not be able to describe fully the roles of hydrolysis and digestion, but basically you can start thinking, what is hydrolysis playing? Well, digestion is breaking down. Hydrolysis is a catabolic reaction, is breaking down. So what can it offer to digestion? So real quick, the answers, you have the monodi and polysaccharides, as well as these main examples. Glucose is a monosaccharide, this is a disaccharide, polysaccharide. And then for digestion is the breakdown of molecules. It makes food soluble. That it allows, which basically means that it can be absorbed in the blood and used. Polymers cannot be used by the body. If I gave you some starch, you would not be able to use that in its form. So basically, polysaccharides are broken down into monosaccharides via hydrolysis. Lipids are broken down into fatty acids, and proteins, which we'll learn next, is broken down into amino acids. Thank you.